All right, week 5. So it's kind of a busy week. You've got less lecture videos to watch than normal, so that's the good news. It kind of balances out a little bit. All right, so the busy part is exam 3 is going to be Monday, Tuesday, and Wednesday, so February 8th through the 10th. I know that's a really quick turnaround because we just had exam 2 last week. Remember, this is on chapters 6, 7, and 8, and those were shorter. So number 1, it's not as much material like length of lectures-wise. Number 2, because it was such a quick turnaround, usually tests are only open for 2 days, like Monday and Tuesday. I'm going to leave this one open through Wednesday. That way that gives you a little bit more time to prepare. Another thing, since I'm leaving it open through the middle of the week, you were supposed to do chapters 6, 7, and 8. There was a review quiz for chapters 6, 7, and 8. And in the announcements and everything, it said that it was due Sunday, February 7th. I'm going to go ahead and leave that open until Wednesday. So that gives you a little bit more time to do it. So if you didn't get a chance to do it before Sunday and you're seeing this, you still got until Wednesday to do that review quiz. So go back and do that if you haven't done that already. All right. We know it's very fresh memory of taking exam 2, so you know how that goes. Just a reminder, make sure you are following those instructions with your environment video. And when you're done with that video, you got to go straight into the test. You can't record your environment and then like walk off for a little while, because that defeats the purpose of showing your environment if you don't take the test right after that. All right, so just a quick note about that. Okay, once you have taken the quiz, or sorry, taken the exam, then we are going to watch the chapter 9 lecture. This one is on the cell cycle, and we're going to focus on something called mitosis. This is how your cells reproduce. All right, so just that one this week, because like I said, you had a test, and next week, good news, we will not have an exam next week. So we'll have time to finish up the lecture material on the exam 4 next week. So just do chapter 9 this week. You've got a review quiz, which is February 14th. We have a discussion board on photosynthesis, so that goes along with the chapter 7 that we did. This one's a little different. I've got a couple articles that I want you to read. And instead of leaving a reply and responding to another student, you can still do that if you want to, but to get your points, I've got 2 different articles. One is on a synthetic leaf. It's like a leaf, man-made device that goes through photosynthesis to make energy. The other one is on aphids who can do photosynthesis. So this is actually an animal that can do photosynthesis. So read both of those, and I want you to leave a comment in both of those articles. So I'll have 2 different threads, one for article 1, one for article 2. Just leave a comment in both of those threads. Okay? All right. Lab this week is also on photosynthesis. So this one, the good news, you don't have a step where you have to like incubate anything overnight like we did last time with the sliced up potatoes. But this one does take a little bit of time because you're going to have to take spinach leaves. So look at the shopping list. Make sure you've got everything. You're going to need spinach leaves. We are going to punch holes or cut squares out of the spinach. Read the instructions. It'll tell you how to prepare this. But we're going to do something to the spinach leaves so that they sink in the water. And then we're going to shine a light on them and allow them to do photosynthesis. So this is cool. The way this works is that when plants do photosynthesis, they produce oxygen. We know that from chapter 7. So if they're sunk in the water, when they start to do photosynthesis because of the light, that's going to produce oxygen, which is a gas. That gas is going to make the spinach disc float up in the water. And that's the way that we can actually kind of prove that photosynthesis is taking place. All right. So the only thing is, the setup of this is pretty quick. It's not too bad. But then you have to set your experiment up. You have to shine light on it. And you have to wait for over an hour. I think a couple hours on one of these. You can be doing other stuff. It's just you're going to have this set up, and every 10 minutes or so you're going to look and notice how many of the leaves are floating. It's not like you have to sit there and stare at the beaker of water for 2 hours. All right. But anyway, all of that to say, make sure that you set aside enough time to do this where you're not going to be distracted and you can make observations when you do this. A couple notes. It says that you need a desk lamp. It says with a minimum 60 watts incandescent, like a screw-in bulb, or a 13-watt fluorescent bulb. I know these instructions are kind of old, and I know that they've changed the wattage on the screw-in incandescent bulbs. So I think if you've got a 43 watt is the equivalent now, that's fine. Just get as bright a light as you can. And that should be okay for this experiment. But you do need a light source, so make sure that you can get one of those lined up if you don't have one already. Okay? So that's it this week. If you have any questions, let me know. Those are all due Sunday, February 14th. It's also Valentine's Day. You probably don't want to spend your Valentine's Day night doing a photosynthesis lab. So feel free to do this early and get it out of the way so you don't have to worry about it on the deadline. All right? If you need anything, let me know, and I'll be glad to help.